Welcome to ICU Primary Prepcast.
Hi, I'm Maddie.
Hi, I'm Swapnil, and welcome back to yet another primary snippet. And this time we're going to talk about receptors. So, Maddie, can you describe what are receptors and also discuss the relationship between the properties of a drug and potential receptor response under the following headings: agonist, partial agonist, inverse agonist, and antagonists?
So receptors are large protein regulatory molecules which natural ligands or drugs bind to, and this results in a biochemical or physiological response. And there are two main types: internal receptors or cell surface or transmembrane receptors. So internal receptors, as the name suggests, are intracellular and they're found in the cytoplasm of cells. They respond to hydrophobic ligands that can travel across the plasma membrane, bind to the receptors, which then causes a conformational change. This exposes a DNA binding site on the protein, and then the ligand receptor complex moves into the nucleus, binds to regulatory regions of chromosomal DNA, and promotes initiation of transcription. So an example of these, steroid receptors. The other major type are cell surface or transmembrane receptors, which bind to external ligand molecules. And the receptor spans the plasma membranes, so ligands do not have to then enter the cell. The cell surface receptors consist of an external ligand binding domain or an extracellular domain, a hydrophobic membrane spanning region, and an intracellular domain inside the cells. And there are three general categories of these kind of receptors, which are ion channel linked, G protein coupled, and enzyme linked. So ion channel link are those where the binding of the ligand to the extracellular domain confers a conformational change in the structure that opens the channel through the membrane, and this allows for specific ions to pass through. So an example of this are GABA receptors. Then there are G protein coupled receptors, which there are huge numbers of, and these have 7 transmembrane domains, and a ligand binding results in activation of a G protein, which then triggers a cascade of intracellular signaling, and this causes a response. So an example of this are beta-adrenergic receptors. And finally, there are enzyme linked, where a ligand binds and there's a signal that's transferred through the membrane, which activates an enzyme, and there's then a chain reaction within the cell, resulting in a response. And an example of this are insulin receptors. So then we think about the relationship between the properties of a drug and the potential receptor response. And before we do this, it's important to define a few terms which will be relevant. So efficacy is a ligand's maximal possible biological response when bound to the receptor, and this reflects the intrinsic activity of a drug, which is the max possible response of the drug when compared to a full agonist. And the effect of the drug is plotted against the log dose, and plateau of the curve is the Emax. And the greater the Emax, the greater the efficacy. Then there's the affinity, which is the extent to which a drug binds to receptors at any given drug concentration. And this can be measured by the concentration of drug that occupies 50% of available receptors, or the KD. So a low KD means that a lower concentration of drug is required to occupy 50% of the receptor, which means that there's a higher affinity of the drug. The potency is the measure of the amount of drug required to produce a response of a specific magnitude. So this is usually measured by the EC50, which is the concentration of a drug that causes 50% of maximum effect. So as you can imagine, then the smaller the EC50, the more potent the drug, as there a smaller amount of drug is required to cause then a specific effect. The potency depends on the affinity of the drug and the efficiency with which the drug receptor interaction is coupled to response. So when we think about the effect of these drugs, there's something called the two-state receptor model, which states that the receptors exist in an equilibrium of inactive and active conformations. So the inactive form does not produce a response, while the active form produces a response. And certain receptors also have constitutive activation, where there is a baseline effect despite no ligand being attached to the receptor. The affinity of a drug determines its interaction with either conformation and its efficacy in producing a response. So then let's come to the different types of response that can occur. So an agonist is a ligand which binds to the receptor and produces a biological response. A full agonist has 100% efficacy or an intrinsic activity of 1, and it has a very high affinity for the active conformation of a receptor. And so the equilibrium is shifted to the active receptor form and a response is generated. A partial agonist is a ligand which binds to a receptor but does not result in the maximal potential response, even if there is full receptor occupancy. So partial agonists therefore have decreased efficacy or an intrinsic activity of between 0 and 1. They have only slightly higher affinity for the active conformation of the receptor compared to the inactive conformation of the receptor. So there's a sub-maximal response. An inverse agonist is a ligand that binds to a receptor but produces the opposite pharmacological effect. So it has an intrinsic activity of between 0 to -1. It has a higher affinity for the inactive receptor conformation. So in those receptors that do exhibit constitutive activity, it reduces the constitutive activity of the receptor as there's less active receptor conformation. And then an antagonist is a ligand that binds to the receptor but produces no response. It therefore has an intrinsic activity of 0 and has an equal affinity for the inactive and the active receptor conformation. So competitive antagonists bind to the same site as agonists, but do not activate the receptor. They shift the agonist dose response curve to the right, but does not change the maximum response of the agonist. So therefore, it decreases the potency of the agonist, but not the efficacy or the maximal possible response. Non-competitive antagonists bind to an allosteric or a non-agonist site on the receptor, but still opposes the action of the agonist. They have an intrinsic activity of 0 and will decrease the efficacy of the agonist, therefore reduce the EMAT. And there are some diagrams in the show notes which kind of illustrate these concepts a bit more clearly. So just have a look at the show notes.
Thanks, Maddie. So receptor is a very favorite topic for primary examiners. And again, that can be asked in various different ways. There could be a specific question around GABA receptor, opioid receptors, or NMDA receptors. And then another way to ask why is around drawing those dose response curves and getting explain how agonists, partial agonists, or partial antagonists work. For example, morphine, buprenorphine. The why can again go into a lot of what are the different types of antagonism, like physiological antagonism or chemical antagonism, for example. And obviously, examples are expected from the good candidates. There are a couple of tricky questions that recently some of the candidates asked me is why the dose response curve, they use a logarithmic scale. And again, that's because the effect of the small dose can result in a large response. That's why the scale is logarithmic scale, not a linear scale. And how the each effect of the antagonist on the curve. So you will find those graphs in the show notes that Maddie has provided, and that will help you to understand the receptor antagonism. So thanks, Maddie. Thanks for all the hard work. We'll be back with another snippet in week's time. Till then, goodbye and have a nice time.
Thanks for listening. See you next time.